Greetings from Krishna IVF Clinic. In today's talk, I'll be speaking about the role of male as a cause of recurrent implantation failure. Even though we have been using IVF ICSI for the last 40 to 50 years, still we have not understood and properly evaluating the role of male factor infertility. The role of sperm in embryo development is crucial, but most of us think after ICSI, the role of male factor does not exist. In this lecture, I'll be taking you through a journey about the effect of sperm on the embryo from the fertilization point to the blastocyst. Generally, when we look at a sperm, we just look at the head, which contains nucleus, capped by acrosome. The neck contains two centrioles, which were earlier thought was only one centriole, but recent evidence suggested that there are two centrioles. The tail, which contains the flagella. But actually, there's more than that to the role of male factor on embryo implantation and outcomes. Once the zygote forms, it is a totipotential cell wherein before it undergoes goes into a diploid zygote, the following events happen. One is oocyte cortical reaction, activation of protein synthesis, resumption of meiosis, and exclusion of the polar body. Here in this video, I will be showing you the morphological events that happen during a time lapse wherein you are seeing the cortical wave. Once you have moved from the cortical wave, the second event is the cytoplasmic wave. The cytoplasmic wave represents the morphokinetic manifestation of the assembly of the microtubule of the spermaster. If the cytoplasmic wave is not proper, it represents that the aster formation is not proper. In fact, a lot of studies are showing that cytoplasmic wave is a good predictor of blastocyst and implantation. So when the aster formation is not proper, then the cytoplasmic wave is not proper, which in turn affects the quality of embryo and implantation. In a physiological situation, the aster enlarges radially from the sperm centriole throughout the zygote cytoplasm, building the cytoskeleton infrastructure through which the two pronuclei are repositioned at different points. This repositioning of the two pronucleus is called PN migration. The pronuclear migration during the actual process of pronucleus migration, as you see in the video, here you are seeing the two pronucleus migrating from periphery to the center. What happens at the cytological level during pronucleus migration? In pronuclear migration, the sperm centrosome splits into two halves, from which two daughter centrosomes are formed and are repositioned at both ends of the meiotic spindle. At the same time, during the migration, the paternal and the maternal chromosomes undergo duplication. The next step is the pronuclear breakdown. Finally, after the alignment of the pronucleus, the pronuclear breakdown happens, during which a sigma aqua occurs, marking the final event of the fertilization and the beginning of the first meiotic division. Here, the role of sperm can act at the entry of the sperm, which produces a calcium oscillation, at the formation of the centriole and the spindle formation, at the formation of the syngamy, and then the correct alignment of the cells. All these play a role, and male factor plays a role at this point. The next major step occurs at 4 to 8 cell stage of the embryo. That's when the male genomic expression starts to begin. Till that time, it's the maternal genome that is most active, and by the time it reaches 4 to 8 stage, the maternal genome transcription and protein activity declines, and the paternal genome has a major role in the embryo development. What is the role of cytoplasmic sperm factors in embryo development? They can be essentially classified into sperm activating factor and centrioles. I'll be talking about the sperm activating factor. The sperm activating factor triggers the signaling cascade of the oocyte activation through cyclical release and uptake of calcium from the endoplasmic reticulum. Happens through a testicular specific isoform phospholipase C, also known as PLC zeta. And PLC zeta can cause oscillation of the calcium and activation of the embryogenesis. Initially, it was thought that PLC zeta has role only at the time of fertilization, but newer evidence is showing that PL zeta abnormality can affect the fertilization, also affect the embryo quality. And last but not the least, there is an association between failed expression of PL zeta and male idiopathic infertility. When we come to this centriole, the centrioles are paternally inherited, and each human sperm contains two remodels per centrioles. Centriole abnormalities cause fertilization failure, delayed or aberrant cleavage, and embryonic, as reported by Tesarik in 2002. Centromere dysfunction and consequently spindle abnormalities may induce defect in the microsome segregation, leading to developmental abnormalities, which such as fragmentation of embryo, abnormal cleavage. So, in the cytoplasmic factors, you have the sperm activating factor and the centriole. When we look into the genetic and epigenetic factors that influence the genetic and epigenetic sperm factors that influence the embryo development, they can be classified into sperm DNA fragmentation, sperm aneuploidy, sperm epigenetic modification, sperm mRNA, sperm methylation. These are the genetic and the epigenetic factors which play a role on the embryonic development. The first thing I will be discussing you about is sperm DNA fragmentation. Here there is a deficiency in chromatin packing, there is an abnormality in the oxidative stress, an abnormality of the apoptosis, which in turn causes damage to the embryo, and its expected effect is mostly at the 4 to 7 embryo when the zygote genomic expression starts. And this phenomena is also known as late paternal effect, as described in Tesarik in 2005. When we come to sperm aneuploidy, maternal aneuploidy is common and it is age related, whereas paternal aneuploidy is not age related. Maternal aneuploidy most of the time affects the non-sex chromosomes, whereas paternal aneuploidy affects the X and the Y chromosomes. The sperm contribution to the embryonic development has remained an enigma. The male zygote appears to be mainly involved in the generation of embryonic sex chromosome and leading to aneuploidy. Male with altered karyotyping are more prone for generating abnormal spermatozoa with aneuploidy. In other words, sperm aneuploidy involves most of the time abnormalities of sex chromosome and also to a certain extent unbalanced chromosomes in the parents. Whereas in a maternal aneuploidy, the common chromosomes that are involved are the 13, 18 and the 21 chromosomes. What are the effect of epigenetic factors? What is the meaning of epigenetic factor? A genetic expression without the actual change in the genomic material is called epigenetic. These can occur in the sperm by the following mechanisms. One is there is a specific methylation profile on the sperm, the effect of protomization of the sperm chromatin, modification of the retained histones after protomization of the sperms, and the complex population of RNA all contribute to the epigenetic signature of the sperm, and these have a profound effect on the embryo. When you look at the architecture of the sperm chromatin, histones are replaced by protamines, and around 10 to 14% are retained histones which can undergo methylation, phosphorylation, acetylation, and abnormal protamization is associated with abnormal morphology. So the architecture of chromatin and understanding the chromatin diagnostic methods like the chromomycin A3 and aniline blue, which we check for chromatin abnormality, which are not picked up by sperm sperm SCDASS. When we look at sperm methylation, these sperms, till they're activated, undergo hypermethylation. The meaning of hypermethylation is the genetic expression is silenced, and once they reach 4 to 8 cell stage, the hypermethylation is stopped and the true influence of the genome is expressed, and sperm methylation patterns are predictive of embryo quality. When we come to the mRNA, the human sperm, when it's packed into a tightly compacted nucleus without any cytoplasm, with a neck and a tail, the sperm becomes transcriptionally and translationally inert, and there are a set of sperm RNA which facilitate in the zygote formation and early embryonic development, and the molecular developments remain unclear. What is the clinical implications of whatever I spoke in the last 10 minutes, which I'll be elaborating? The sperm and the clinical outcome on an IVF depends on the quality of the sperm. The significant impact of oocyte on embryo morphology has overshadowed the sperm effects, and only recently now it is becoming more apparent that more than this oocyte, the sperm is equally important in the development of the embryo and the implantation and the ongoing pregnancy. If you look at this slide, the total sperm count, which is correlated with the number of oocytes, the blue one is the 1 to 5 oocytes, the red one is 6 to 10 oocytes, and the green one is more than 10 oocytes. Here you see the relation between total sperm count and the number of oocytes. When the total sperm count is good and the number of oocytes are good, it really doesn't make a difference. But its impact on total sperm count, when the oocytes are few, it really makes a difference. So this helps in counseling a patient. If you have a patient with low ovarian reserves or a poor ovarian reserves with a poor male factor, then the total sperm count is abnormal, and there you have a difference in the outcome. This is the impact of total sperm count. Similarly, when we come to morphology, there is a beautiful paper which was published in 2018 where evaluation of impact of sperm morphology on aneuploidy rates in a donor oocyte program. Here what they did was they used a cohort of oocytes and used different morphologies to study the quality of embryo formation and aneuploidy. And the study investigated the correlation between sperm morphology and the incidence of embryo aneuploidy in a oocyte donation program. This data has showed that when the abnormal morphology is very high, even in a donor oocyte program, the quality of embryo gets affected, and suggesting that for implantation failure or suboptimal embryos or recurrent implantation failure, looking at sperm morphology can play a major role. When we come to the role of paternal age, when there is a significant difference in the male factor age more than 40 years compared to the woman, there is an additive effect, and if both the partners are of higher age, there is an additive effect on the pregnancy and live birth, and both the partners are at advanced age. So there is a role for in advanced stage that the IVF outcomes will be different from where the partners are lower age. This slide summarizes the effect of different parameters of whatever I told. This graph shows PL zeta. The intensity of the color shows how much it is active at the oocyte to the embryo level. Then the second column is the centriole. The third column is the DNA integrity. The fourth is the chromosome complement system. The fifth is the chromatin architecture. The sixth is the DNA methylation. The seventh is the RNA. The PLS zeta triggers a mechanism of cyclical oocyte oscillation of the concentration of the intracellular DNA, and this plays a major role in fertilization. The centriole directs the organization of the oocyte centrosome, which regulates the pronuclear position and assembly of the mitotic spindle during the first cleavage. The integrity of the sperm DNA influences the embryo quality at the early and the late stage of blastocyst development. Sperm chromosome abnormalities which affect 4 to 5% of all pregnancies, and sperm chromatin affects embryo quality at cleavage stage, and dysregulation of sperm methylation is associated with reduced embryo quality. Sperm derived mRNA are present in fertilized oocytes and their presence decreases over time during the development stage, but their protein products are present at the blastocyst stage. And these are the 7 factors that affect the embryo. The 7 factors that affect the embryo are PLC zeta, the centriole, the DNA integrity, the chromosome complement, the chromatin architecture, DNA methylation, and the RNA. In conclusion, I wanted to emphasize, rather than the clinical aspect, the biological aspect of the effect of males on the embryo quality and the implantation. I thank one and all for giving this opportunity.